Hi, my name's Harmon. I'm a fourth-year undergraduate student at the University of Wisconsin-Madison. I'd like to thank the organizers for inviting me to give this talk. Today, we'll discuss a new matrix factorization method for cell type identification. But first, let's start with an overview. Single-cell genomics has revolutionized our capabilities of understanding complex biological systems and structures. It has enabled several applications in understanding cell behaviors, including fate decisions, developmental transitions, and responses to disease. One problem that is common to many application areas is cell type identification. Let me introduce you to that. Given a group of cells, we'd like to identify their corresponding cell types. The two most popular approaches for this are supervised and unsupervised learning. While supervised learning approaches are promising, they require the transferring of labels from labeled to unlabeled data sets. These labeled data sets may not exist in cells from disease states, rare tissue, or rare organoids. Therefore, we'll focus on the class of unsupervised learning methods. A common workflow for this question is as follows. We start with a single-cell RNA sequencing dataset, which you can represent as a cell-by-gene matrix. First, we do dimensionality reduction to bring the data into a lower dimensional space. Second, we do clustering to group cells with similar representations. Lastly, we can annotate cell clusters using curated marker gene databases. To do so, we have to identify genes for each of these clusters. The most popular approach is differential expression analysis. However, it lacks specificity, and more importantly, the cells are not informed by the genes. The latter is important because DE genes may not be the genes that are driving the feature representations, and DE analysis may have a difficult time of identifying shared genes, especially in contexts where cells are similar. We'd like a way to cluster cells and gene to cluster cells and gene programs simultaneously. This is where non-negative matrix factorization comes in. Non-negative matrix factorization, also known as NMF, aims to optimize this objective right here. The main parameter here is K, which allows us to choose the number of factors that we'd like to represent our lower dimensional embeddings by. We can use the lower dimensional embeddings of the cells and genes to identify cell clusters along with their corresponding gene clusters by leveraging the factors. These cell clusters map to gene clusters in a one-to-one fashion. However, this is too restrictive. How can we solve this problem? I'd like to introduce you to Scotch. We introduce a new method which is based on non-negative matrix tri-factorization, also known as NMTF. Whereas NMF only allows for a one-to-one mapping between cell and gene clusters, Scotch allows for a many-to-many mapping. This is possible because of the additional S matrix. This many-to-many mapping grants Scotch two large advantages over NMF. First, we can choose different numbers of factors for both spaces, indicated by K1 and K2. Second, we can regularize both the cell and gene space simultaneously. This is not possible in NMF as it leads to over-regularization. In Scotch, we implement orthogonal regularization to encourage a stronger factor structure. Thus, U gives us the cell representation, V gives us the gene representation, and S tells us how they interact. Similar to NMF, we assign cells and genes to the factors that best represent them. We use the S matrix to understand how cell clusters map to gene clusters. We then annotate gene clusters with terms from curated marker gene databases after running an enrichment test. We look at cell type enrichment, which is represented by the red circles, and gene ontology enrichment, which is represented by the blue circles. We assess Scotch's performance by evaluating it on three data sets. The first data set is a simulated data set where the number of true cell and gene clusters are known. Just to be clear, we don't actually have cells and genes in this data set, but rather rows and columns. I use the term cells and genes to keep a consistent naming convention across the data sets. The second data set is a well-known single-cell RNA sequencing data set used for benchmarking, PBMCs. In this data set, we know the true cell clusters, but we do not know the true gene clusters. The last data set consists of cells from 2D and 3D retinal organoids. Neither the cell nor gene clusters are known in this data set. Let's start with the simulated data set. To test if Scotch works, we simulate a data set where the cell clusters map to gene clusters in a many-to-many fashion. Additionally, both the cell and gene spaces have different numbers of clusters. With this in mind, we can now look at how Scotch performs compared to ONMF, which is non-negative matrix factorization with orthogonal regularization in the cell space. To measure accuracy, we use adjusted RAND index, also known as ARI. ARI computes a score that tells us how similar predicted clusters are to the true clustering. A score of 1 indicates perfect prediction of the true clusters. The x-axis is the cell cluster ARI, whereas the y-axis is the gene cluster ARI. Each point represents the score of a specific choice of hyperparameters over all seeds. We can see that Scotch is able to outperform ONMF in this domain, which represents the optimal ARI for both the cell and gene clustering. Now that we've shown that Scotch can do what we promised, we apply it to a single-cell RNA sequencing dataset. PBMC datasets are commonly used for benchmarking. This dataset has B cells, T cells, monocytes, natural killer cells, dendritic cells, and megakaryocytes. We use this dataset to benchmark Scotch against ONMF and Louvain clustering, as we have the true cell clusters. To the right, we have the ground truth cell cluster labels. And to the left, we have the cell cluster labels for all of the different methods. We've projected them using UMAP. To get a better understanding of how they compare, we can look at a cluster composition heat map. There are a total of 8 true cell types. Scotch and ONMF under cluster, only pulling out 6 clusters, whereas Louvain over clusters, pulling out 9 clusters. Since it can be kind of hard to understand which method clusters better by just looking at the heat maps, we again introduce the ARI score. What we can see is that Scotch performs the best. While cell cluster accuracy is important, we're more interested in our ability to annotate cell clusters, and to do that, we have to look at enrichment. The way we read this plot is by using a bottom-up approach. Starting at the gene cluster enrichment, we look for terms that we may be interested in looking at. In this case, it's effector CD8 plus memory T cells. We can find the corresponding gene cluster that has the most enrichment and trace it up the plot. Once we do this, we hit the S matrix. In the S matrix, we look for darker boxes, which indicate the corresponding cell clusters for that specific gene cluster. And what we can find is that cell cluster 6 connects to gene cluster 14. It's predominantly CD8 T cell, and we can also see that it has a decent amount of natural killer cells in it, which we also get enrichment for in gene cluster 14. You may have noticed that there are other gene clusters that connect to cell cluster 6. If we look at these gene clusters, we can see that we get additional enrichment for Go terms as well as natural killer cells. The nice part about Scotch is that we're able to pull out specific gene programs. For example, antigen processing via MHC class 1 is a function that is specific to CD8 T cells. Now that I've shown you that Scotch can pull out specific gene programs, it's time to compare it to Louvain and ONMF. Looking at ONMF first, you can see that we get less enrichment than Scotch, making it more difficult to annotate the gene clusters, the cell clusters. Louvain, on the other hand, gets too much enrichment, also making it difficult to annotate these cell clusters. Now that we've seen that Scotch can extract biologically meaningful gene clusters in well-studied data set, we'd like to see how it performs in a novel data set. We apply Scotch to a retinal organoid data set with samples from 2D and 3D cell cultures. Gene editing is more tractable in 2D cells. However, the dissociation process from 3D to 2D causes different cellular phenotypes to arise. We're interested in identifying these phenotypes using Scotch. My mouse is frozen. Oh, it's there. Okay, I did not notice this. Yeah, so at the top, we have the cell cluster assignments for Scotch. And at the bottom, what we've done is label that same UMAP with the batch samples. To get a better understanding of how our cell clusters compose of the different batches, we can look at the cell cluster composition heat map again. What we find here is that we get some cell clusters that have a mix of 2D and 3D samples, whereas the rest of them are heavily skewed towards either 2D or 3D cells. To annotate the cell clusters, we now have to look at the S matrix and the gene cluster enrichment again. Okay. Now that we're all set up, let's deep dive into our analysis. Okay, yeah, so what we can see here is that we get enrichment for RPCs in 2D samples. RPCs, also known as retinal progenitor cells, are cells that later differentiate into photoreceptor cells. However, since we get enrichment for these RPCs, it means that these cells were stopped in their differentiation towards photoreceptor cells. Okay. While some of the 2D samples did not make it to their photoreceptor states, we can see that some of the 2D samples do. Here we get enrichment for both rods and cones in a 2D dominant cell culture. This indicates that the gene programs necessary for photoreceptor differentiation exist in the 2D samples, but are probably not in abundance. In summary, Scotch was able to pull out differences between 2D and 3D retinal organoids. With that, I'm going to wrap things up. This is my joint work with Spencer Hallberg Spencer, so I'd like to thank him for his major contributions in developing the method and helping me along the way. I'd like to thank everyone at our lab at the Wisconsin Institute for Discovery, especially our advisor, Shushmita Roy. I'd like to thank our collaborators for generating the data and the NIH and McDonald Foundation for funding this project. At this point, I'm willing to take any questions that you may have.
So is Scotch a new type of integrative ONMF, or have you compared it to like other versions that also did many-to-many ONMF methods like IONMF in 2016?
Repeat the question.
Oh yeah. So the question was, is Scotch a completely new method, and have we compared it to other methods that do many-to-many clustering? So we are, yeah, this is a new method. So we're based off of non-negative matrix tri-factorization. That paper was originally released in like 2001 with Word documents, but we add in orthogonal regularization and we develop our own unique algorithm for like solving the problem. So in that aspect, it's new. To my knowledge, like I guess at MTF or like this sort of algorithm hasn't really been applied to like omics data sets. Like there are some other integrations of like many-to-many mappings, but it's more so for drug discovery. But yeah, we did not compare it to any other many-to-many mappings. Our goal is mainly to show that like NMF is used very often, so is Louvain clustering. We don't think that these are like the best methods, and we'd rather have you use like our method over that because again, like the true biology probably has like, you're probably sharing many gene programs between cell types that are similar and even dissimilar. So with our method, you're able to capture that while also being like highly interpretable. Yes. Yeah.
Okay, I'm not sure if I missed it or so like in NMF, like we have to choose a number of factors. So like in your method, so how do you choose the number of factors, or like is there a protocol to do that?
Yeah, so this is actually so in the data sets where we have adjusted RAN index, we can just look at that and select the parameter that scores the highest in that. But in the other data sets, we kind of have to use other clustering metrics. So first of all, we sort of do like this grid search. So we're trying to we'll kind of choose hyper parameters that we think could be reasonable. So for the data set with PBMC, there were 8 cell types. We kind of looked at like the numbers from 2 to 8, and we did that for both K1 and K2. And then we also just did like this grid search for the regularization terms. But what we can actually do is, we found that the cell adjusted RAN index was correlated with the cell silhouette index, which is also another cluster accuracy metric. So what we could do is this sort of grid search where we can just like plot out the accuracy scores for the method over different hyperparameters. And once we start getting closer to hyperparameters that we like, we might make like more fine-grained changes. But yeah, since it's an unsupervised learning method, there isn't like this. Yeah, this is up to the interpretation of the person who's using it. But I think like you could choose different hyperparameters based on how large the data set is, and also like how many cell types you think might be there. Or so it's not automatic. That's the only bad part. Yeah.
So my question would be whether you will be able to achieve any raised populations that are not studied or defined by other methods or any supervised methods. Still, we are getting these ranges. So as you know, they have very low markers.
Yeah.
Get to the raised populations like or mate or entity versus mate versus entity. Can you get that fine granularity in their populations?
All right, so let me just try to restate your question. So you're asking if we can identify non-common cell types using our method? Yeah, so in the case that you have specific marker genes that you think may be associated with these like traits, the nice part, so I also have an analysis of this, but let me find this. Okay, so here, this loads. Yeah, you can essentially, so since you have gene clusters, you can look at the corresponding gene clusters and what genes exist in that cluster. And if you have, let's say, like a biologist who thinks that like this gene might be associated with a specific type of phenotype, you can kind of say like, oh, this cell might be coming from this sort of disease state. Like I do understand that, like our ability to do actual annotation is going to be a little more difficult, but since we're clustering the genes and putting it into distinct sets, if you do know that specific genes are associated with this rare cell type and you find them all in a gene cluster, it might be safe to assume that, okay, like this might be correlated with this like very rare cell type that you have. I don't know if that answers your question.
Okay.